The cell is the smallest unit of life. Since we are a multicellular organism, we're composed of a number of cells. That means our cells had to go through a cell division process. That process is known as mitosis. There are three major parts of a cell. The cell membrane acts as the boundary of the cell. It controls what enters and leaves the cell. In the center of the cell is the nucleus. The nucleus is surrounded by its own membrane. In the nucleus is where the DNA and the chromosomes are found. The DNA controls everything the cell can do, so the nucleus is the control center of the cell. Then between the nucleus and the cell membrane is a gelatinous type of material called the cytoplasm. The cellular organelles are located here. This is where all of the chemical reactions that occur in the cell happen. If we look at the life cycle of a cell, something we can call the cell cycle, it's divided into two major parts. We have interphase. Interphase is composed of three parts, G1, S, and G2. During interphase, the cell is simply being a cell. The cell is growing. As it goes into the later stages of interphase, it begins to make preparation for cell division to occur. The M phase is divided into two major parts, mitosis, which is when the nucleus divides, and then cytokinesis, which is when the cytoplasm and all the cellular organelles divide. If we look at a cell in interphase, during the G1 phase, the cell is growing. It's doing whatever it does as a normal cell. The S phase is the replication of the DNA. Once the cell enters the S phase, it is going to divide. Now, some cells get stuck in this G1 phase and never divide. Heart cells when you're an adult, your nervous cells when you're an adult, these fail to divide. They stay stuck in G1. But the other cells in your body are capable of going into this S phase. In the S phase, we make a copy of the DNA. That's the genes and the chromosomes. When we have duplicated the chromosomes, the duplicates are held together by a centromere. These duplicated chromosomes that are held together by the centromere are technically called chromatids. All a chromatid is a pair of duplicated chromosomes. The two chromosomes are identical to each other. The G2 phase of interphase is when the organelles divide. If we're going to divide the cytoplasm, we're going to have to make sure that there are enough organelles to go into two cells. When you see a cell in interphase, you can see the cell membrane, you can see the cytoplasm, and you can see the nucleus. And the nucleus looks fairly smooth inside. Those chromosomes are coiled tightly and they don't show up as bar-like structures. The first stage of mitosis is prophase. During prophase, the chromosomes, and remember these are really chromatids that are held together by centromeres, become visible. Notice that the nuclear membrane is gone, and we sort of see these bar-like structures that are those chromatids or chromosomes. As we go into the next stage of mitosis, or metaphase, we have all of those chromatids that line up in the middle of the cell. We have special structures called centrosomes. These centrosomes make spindle fibers. The spindle fibers help anchor the centrosome to the inside of the cell membrane, and they also attach to the centromeres on each of those chromatids. In anaphase is when the centromeres divide. We now have full-fledged chromosomes. The duplicates separate because they're no longer held together by their centromeres, and these spindle fibers begin to shorten and pull the duplicated chromosomes to each end of the cell. In telophase, we begin to see the cleavage furrow. Now, this is the beginning of cytokinesis. The cleavage furrow is where the cell starts to sort of pull apart, and we sort of get cell membrane forming here. The nucleus begins to reform. We get our nuclear membrane back, and those chromosomes become invisible again. The spindle fibers disappear, and as the cell continues cytokinesis, we end up with two cells exactly like the original cell. Cytokinesis is the very last part of the M phase. Mitosis, therefore, produces two new daughter cells. Both cells are identical to each other, and they're identical to the original cell. Both cells have some new parts and some old parts in their contents. The cell then goes through interphase. It grows until the next cell division process. The purpose of mitosis is for growth and repair. Because we make two new cells, it allows an organism to grow, and if there is damage, it allows us to replace the damaged area with cells just like the ones that were there before.